So this is a match question. So whenever you have match question, keep an eye on the options. Make the first match and cross check with the options. So complex 1 is NADH dehydrogenase. A is 1. So rule out option 1 and option 3. So now complex 2 is succinate dehydrogenase. B4. Where is B4? It's in option 2. So that means option 4 is also incorrect and we have the answer with us. Yes, so option 2 is the right answer of this question. ETS complex 3 is cytochrome C oxidase. No, it is BC1. So this is also known as cytochrome C reductase. So C is 2. C is 2. Yeah, C is 2 here. And complex 4 is oxidase, cytochrome C oxidase. This is 3. So let us understand this ETS chain. So ETS is present in which organelle? This is mitochondria, the powerhouse of the cell. So mitochondria is a double membraned structure. So the inner membrane is folded like this and it is called cristae. So this increases the surface area for the ETS chain. So let us familiarize ourselves with some terminologies. This outer membrane is OM, outer membrane. The inner membrane is IM. So what is the name given to the space between OM and IM, outer membrane and inner membrane? The space is called intermembrane space. And what is this part called? The fluid part of mitochondria. This is called matrix. So ETS are present on the inner membrane of mitochondria. So in the textbook, you will find a figure like this. So they have shown this is the inner membrane. This side of the inner membrane is labeled as matrix, and this is the intermembrane space. This is intermembrane space, and this is the outer membrane. Outer membrane. So why I am drawing this? So that you get an idea of exactly what terms are used in the textbook. So matrix, Krebs cycle occurs here in the matrix. TCA cycle. It's not shown in the diagram given in the textbook. So in case, just to revise the and see the whole process in totality, I am drawing this also. So this will give you an idea that this is the source of NADH plus H plus. So NAD carries the hydrogen and it donates it over here. So first complex is called, this is complex 1. It is called NADH dehydrogenase because it removes hydrogen from NADH. Therefore, it is called NADH dehydrogenase. So what will happen? NADH is going to donate and become NAD plus, and it will go back to the cycle, TCA cycle. So You check the diagram, then you will find something FMN written here. These are the flavoproteins. In the text part, it is there's no mention of what FMN is. FMN are flavoproteins. Now the importance of flavoprotein is they are going to accept hydrogen. Okay, they will accept hydrogen, but when they donate it to FES, FES are iron sulfur proteins. So they donate only electron to FES. So what happens to the protons? The protons are pumped here in the intermembrane space. So hydrogen is split into proton and electron. Protons are pumped in intermembrane space and electron goes to FES. From FES, then it goes to, they have again drawn FES here. In the textbook you will find it. And then second complex is succinate dehydrogenase. So succinate dehydrogenase will accept another molecule which comes from TCA cycle, which is FADH2, and it forms FAD plus. So in this case you will find one molecule which is called Q. It's coenzyme Q. So this is basically quinone. So quinone accepts hydrogen. It is like flavoprotein. It accepts hydrogen and it becomes quinol. Quinol, the reduced form of quinone. Quinone is a ketone and it becomes, after reduction, it becomes an alcohol, quinol. So now this is complex 2, and then we have, from here it is going to donate proton and send it to the intermembrane space. The electron is accepted by the third complex, complex 3, and this complex is BC1. So from here the electron travels to cytochrome C. So cytochrome C is mentioned in the textbook. It is on the outer side of inner membrane. So over here cytochrome C. So from cytochrome C, then it goes to the fourth complex, complex number 4. This is called AA3. And from AA3, then finally the electron is accepted by oxygen. So during this process, protons are pumped out. And a portion of protons, they combine with oxygen to form H2O. So we have half O2 and then 2 H plus ions. When they combine, they will form water molecule. So this is how the protons accumulate in the intermembrane space. And then finally, complex 5, which is ATP synthase, is going to accept all the protons and pump them back into the matrix. And during that process, ATP synthesis occurs. So this is all about the ETS, electron transport chain, as given in the textbook. I could have taken any diagram from other sources, but I am trying to fix only to the data and the information which is given in the NCRT textbook so that it makes sense to you while you read it. So the only information which was missing in NCRT is the flavoproteins. In case they confuse you, FMN and Q Remember this way, these two are, they accept hydrogen, but they donate electron, donate electron. And this is the reason when they accept hydrogen, they pump out protons in the intermembrane space and donate electron to the next electron carrier. FES, FES, iron sulfur proteins. And what else? The cytochromes. Cytochromes, they accept electron and they donate electron. Donate. So this information is not in the textbook. So I hope this extra information will make sense to you, how the electrons are moving and why at these steps protons are pumped out. So this is the background behind this electron transport system.